Hola, estudiantes de la biología. Okay, today's screencast is on chemiosmosis, and one of these words should already be familiar to you. You know the word osmosis, it's the movement of water, but in this case we're actually talking about chemiosmosis, which is the movement of chemicals. In particular, we're going to be talking about the movement of H+ ions, or protons, across the membrane. But before we get to chemiosmosis, I want to set up a brief metaphor for you, or analogy, I guess, that will hopefully help make chemiosmosis a little bit easier to understand. So, and the analogy we're going to make is the analogy of a hydroelectric dam. So if you have a dam and you take a stream or a river and you put a barrier in it, that barrier is this thing right here, and you dam up the water, and so you get some sort of lake behind the dam. And obviously the dam is not just a barrier, but there's going to be a place within the barrier that water can pass through. Oh, damn it. Okay, this place that water can pass through, and inside this little tunnel is going to be some sort of turbine that, as water, H2O, moves through this turbine is going to spin, and that's going to power some sort of generator, which is going to be behind it, and the generator is hooked up to a wire, which passes through a transport system and goes to the town. So we are using the power of water to push electrons through a wire, and the reason why this whole system works is because we have an area of high potential energy here and low potential energy here. We essentially have a concentration gradient, so water is going to be moving down its concentration gradient, and that supplies the power to run the generator. However, if we were to take water and run it entirely through, such that there was equal amount of water on both sides of the dam, we'd no longer have this difference in potential energy on either side of the dam. And we're no longer going to be able to generate electricity because we've reached equilibrium. So it's really important to keep in mind that to run a dam, you need to have this difference in potential energy across the dam. Very much similarly, you'll see why this is going to come into play later on when we talk about chemiosmosis. Okay, so let's situate ourselves again within the chloroplast. This is a thylakoid right here, and we've got our electron, or our photosystems, and then our ETSs following the photosystems. And as you remember, light comes in, strikes the photosystems, and excites electrons. And we talked about today in class that one piece of photosystem 2, let's write that here, this is PS2. Photosystem 2 is exciting electrons, and it's grabbing those electrons from a molecule of water that it splits. So water comes in here, and out comes O2 and H+. And we also steal an electron, and that's the electron that's going to be passed down through our ETS and then to our next photosystem. But what I want to talk for you for a second about is this H+. So as this system is running and running and running, we get more and more H+ building up inside our thylakoid membrane because we have this photolysis of water occurring to grab the electrons to run this thing. And we've got a very special protein that I think I've mentioned before, but if not, I'll say it now. This protein is called ATP synthase. And it's also located in, oh, that looks terrible. Okay, that's a little better. So this is ATP synthase. It's located within the membrane of our thylakoid, and the role of ATP synthase is to take ADP, which is adenosine diphosphate, and take a free phosphate group, PI, and catalyze the formation of ATP. Adenosine, oops, that looks like a D. Okay, that's a little better. ATP. Okay, so how does this molecule actually catalyze this transformation of ADP to ATP? Well, it utilizes the concentration gradient within our thylakoid membrane. So notice here that the concentration of hydrogen ions or protons within, and I'm drawing a few more because it's not just in one stack, it's without the whole stack. Okay, the concentration inside the thylakoid is much higher than the concentration of H+ out in the stroma. So there's a pressure for hydrogen to move through or to move outside of the thylakoid, and the only way it can get out is through this molecule, ATP synthase. So just like our hydroelectric dam, where we have a difference in potential energy of the water here as opposed to here, we have a difference in potential energy of hydrogen, and there's a pressure to move out. So water is, or hydrogen, it's going to diffuse down its concentration gradient outside of the thylakoid. Ooh. This one's on its side now. And create, in doing so, it's going to spin a little turbine in this molecule of ATP synthase, and it's going to catalyze the formation of ATP. But we get to this point in the process where now a bunch of hydrogen has moved out through ATP synthase, and we have equal concentration of hydrogen inside the thylakoid membrane to hydrogen outside the thylakoid membrane. So how does the cell deal with this, or how does the plant or protist or bacteria deal with having this equal concentration of H+ inside as opposed to outside? Because remember, it's the driving force behind the movement of hydrogen through this molecule is the concentration gradient. So if the concentration gradient is eliminated, there's no longer a pressure through this molecule. It's just like our dam scenario where water is equal on both sides of the dam. So in order to keep the concentration gradient of hydrogen high inside the thylakoid stack, this, this photosystem excites an electron and passes it down the electron transport system, and this electron transport system pumps hydrogen against its concentration gradient into the thylakoid. So hydrogen is moved back through this ETS, back into the thylakoid, and remember, this is against its concentration gradient, so this is a form of active transport. It takes energy. So this is how the concentration gradient of hydrogen is maintained. Keeps it high in the thylakoid and low outside in the stroma. I'm going to write that here, stroma. Okay, I think that is it for today. I guess the last piece that I should tell you is that another important thing that we have not really talked about yet is the function of the ETS that follows photosystem 1. Well, you know, electrons, high energy electrons are being, the electrons are being excited in the photosystems, and then they're passed down the electron transport systems where they used to do work. In the first one, we pump hydrogen through the membrane. That's the work that's done. And in the second one, there actually isn't a lot of work done. We pass on the electrons to a carrier molecule. It is called NADP+. So NADP+, plus 2 high energy electrons, plus a hydrogen ion, are going to be all stuck together to get NADPH. Now, the important thing to remember about NADPH is that it has 2 high-energy electrons in it. And so these electrons can be used to do work later on. So both NADPH and ATP are created in the stroma, and we're going to talk tomorrow about how they are used to catalyze the light-independent reactions, because everything that we've been talking about photosynthesis up to this point has been all light-dependent reactions. But remember, there's a whole second part of the process that we haven't even touched upon yet. But that is for tomorrow if you are in B block, and also E block as well. Okay, so this is it, and I will see you all tomorrow. Adios. Or adio, as they say in Nicaragua.